Thank you for this nice introduction. Come on. Good morning. My name is Cléo Chidaine. I'm a second year PhD student at the University of Bordeaux in France. And my PhD is supervised by the Dr. Jeanne Leblanchain and the Dr. Laurent Azema. And today I have this great opportunity to present you my side project called nanostructure based on an aptaswitch aptakiss interaction. So of course, for this event, I don't need to introduce you what is an aptamer, but today I just want to focus on two different structures. First, the anti-adenosine aptamer. As you can see, this oligonucleotide can specifically interact with a small molecule, the adenosine, but I also want to introduce you the kissing complex structure. And as you can see, this structure is composed by two stable structures. Here, I want to show you these two structures because in this talk, I want to focus on one specific type of aptamer called kissing aptaswitch. In fact, how can we create a kissing aptaswitch? For example, if we have a sequence that can specifically interact with a small molecule, for example, adenosine, and if we combine this sequence with another sequence that can form a stem loop involved in a kissing complex, we can create a new oligonucleotide that can specifically interact with the adenosine, but also create a kissing complex interaction. So if I want to answer to the question, what is a kissing aptaswitch, I would say it's a dynamic aptamer that can form a stem loop structure depending on the presence or the absence of the target. And as I said, this stem loop can create a kissing complex interaction with a small oligonucleotide that we call aptakiss. So based on this principle, we can have two different possibilities. For the first one, we will talk about a positive aptaswitch because this aptaswitch cannot form the same structure. And if we add the adenosine, so the target, the interaction with the target will stabilize the same structure. And finally, the kissing complex interaction is possible only if the target is present. For the second possibility, here is the opposite. With the negative aptaswitch, this oligonucleotide can always form the same structure. But if we add the target, the interaction will destabilize the secondary structure and finally break the interaction between these two partners. Sorry. So here in our laboratory, we have two different aptaswitches against the adenosine, a positive one and a negative one. These two aptaswitches was characterized by fluorescence anisotropy. I cannot enter into the details, but in fact, with this technique, we can measure the interaction, the kissing interaction between these two partners. For example, if we increase the aptaswitch concentration in presence of adenosine, we can measure a difference on the fluorescence characteristic, and this difference correlates to the interaction between the two partners. Also, there is something that we need to remember. It's the kissing complex is magnesium dependent because if we try to do this experiment without magnesium, we cannot form the stem loop, the kissing complex structure. Also, this system is very specific to the adenosine because if we do this experiment with the adenosine, we can measure this signal. But if we use the inosine instead of the adenosine here, we will lose this signal. The same, if we put a mutation on the kissing complex structure, here, the system doesn't work. Finally, we have a three-component system, and if we use the two oligonucleotides, the magnesium, and if we increase the adenosine concentration, we can increase the interaction between these two partners. For the negative aptaswitch, it's the same principle, except we don't need to put the adenosine to be able to observe the interaction between the two partners. Finally, if we increase the adenosine concentration, we will lose this interaction. So this type of molecules can be used to develop biosensor systems, build supramolecular assemblies, and develop DNA nanomachines. And today I will focus on these two parts. So first, how can we build a supramolecular assembly based on this aptaswitch aptakiss interaction? For example, in 2018, it was possible to my supervisor to create a nanostructure based on the positive aptaswitch. For example, if we use a dimeric form of the aptaswitch and a dimeric aptakiss, and if we add the adenosine, we can form a structure, and this structure can be observed by massive phage or atomic force microscopy. Here in this project, I don't want to use the positive aptaswitch, but I want to create supramolecular assembly with the negative aptaswitch aptakiss system. And here, I want to use this type of DNA cross structure to build these assemblies. The idea is simple. We want to have two different crosses. The cross A is functionalized at its extremity with two negative aptakiss, and the cross B with two negative aptaswitch. And here, the objective is try to see if we can observe an interaction between these two crosses without adenosine, and if we can break this interaction by adenosine addition. Okay, so first, I confirmed that it was possible to build the DNA cross. So with this agarose gel, I can check the folding of the different parts of this structure. And finally, if I put all the strain, I can form one object that corresponds to the cross structure. And after that, I also confirmed that it was possible to build the different parts of this cross. To functionalize these two crosses with the two negative aptakiss or with the two negative aptaswitches. So now we can build these two crosses, and we want to observe an interaction between the two constructions. So to do that, we can perform a native page with or without adenosine. So here, for example, if we focus first on these two crosses only, if we analyze these two crosses, we can observe two bands that correspond to cross A and B. But now, if we mix these two crosses together, here we cannot observe these two bands, and now we have something at the middle of the gel that could correspond to a dimeric form of our crosses. And the most interesting object was present in the well. Here, we always form this object on the well, and this object is too big to enter inside the acrylamide structure. So even if we change the acrylamide percentage or the bisacrylamide acrylamide proportions, this object is always present on the well. And here, it's a polymeric form of our crosses. So if we try to do this experiment again, but this time we also put the adenosine inside the acrylamide, here we cannot observe anymore the polymeric form present on the well, and now we can form again the two bonds that correspond to the construction A and B. So here, with this sample experiment, it was possible to show the interaction between the two crosses and to prove that the adenosine addition can perturb this interaction. In terms of perspective with this part, we want to try to study the polymeric form by atomic force microscopy. And one other idea is try to create a DNA nanogrid. For example, if we use these two crosses functionalized at each extremity, it should be possible to have a structure that can self-assemble spontaneously without adenosine and disassemble in presence of adenosine. Okay. In the second part, we also try to create a dynamic nanomachine, DNA nanomachine, and here the idea is try to have a DNA actuator based on the negative aptaswitch, but also the positive aptaswitch. Here we want to have a system with two crosses, and we want to link these two crosses with a chemical linker. And here we want to functionalize one part of this construction with the negative aptaswitch aptakiss system, and we want to functionalize the other part with the positive aptaswitch aptakiss. Here, if we add the adenosine, we want to induce a conformational change on this construction. So we want to destabilize the kissing with the negative aptakiss and stabilize the interaction on the other part. Moreover, we also want to add a threat reporter on this system. So to do that, we can put a green fluorescent molecule on the negative aptakiss and the cohesion molecule on the negative aptaswitch. And here, if these two molecules are very close in space, we want to observe a threat effect that will turn off the fluorescence. And so if we add the adenosine, we want to observe a change on the fluorescence properties. So again, I checked that it was possible to build the two crosses that compose this actuator. So the part with the two aptakiss or the part with the two aptaswitches. And then I tried to link these two parts with the chemical linker. So here, if I mix these two processes with this linker at the same quantity, I can generate a new object, a bigger object that corresponds to the double cross system. So for this experiment, I did 4 different constructions. The construction 1 is just a green fluorescent cross. The construction 2, it's a double fluorescent cross. The 3, it's the system just with the negative aptaswitch aptakiss. And the 4, it's the entire system. So now, this is what we can observe if we analyze this construction by a native page again. So first, let's focus on the construction 1 and 2. Here we can make the difference between a simple cross and a double cross. So now let's focus on the construction 3. With the construction 3, we can observe a band here with a low fluorescence intensity. And this loss of fluorescence is due to the presence of the quencher. But now, if we add the adenosine, we can observe a change on the migration profile. In fact, this shift on the migration is due to the conformational change of our construction because we pass to a closed state to an open state. And here, this second conformation have another migration profile. Moreover, it looks like the fluorescence intensity increased a bit between these two conditions, but the problem is with the adenosine, we cannot restore completely the fluorescence intensity, and we cannot obtain again a band with high fluorescence intensity, such as the construction 1 or 2. If we focus on the construction 4 now, it's a bit the same result. We have here a band with a low fluorescence intensity, and if we add the adenosine, this fluorescence increases a bit, but again, we cannot restore completely the fluorescence. The interesting point with this last construction is the difference between these two, the difference on the migration is not very important if you compare this migration with the construction 3. And here it makes sense because it's probably due to the conformational change. Here we have a closed state with the kissing in one part, and we pass to another closed state. So maybe it's the reason why the difference is not very important. Just to confirm what we saw in terms of intensity, we also did this experiment, but not in the acrylamide gel, but in solution. And here, if we measure the fluorescence intensity with or without adenosine, we can effectively measure a significant difference between these two conditions. That means that the adenosine have an effect on the fluorescence intensity of these two constructions. Okay, just to conclude with this second part, here we had some preliminary results with this type of constructions, and maybe in the future we will try to complexify these nanomachines. For example, we can try to add some fluorophore and quencher on the positive aptaswitch aptakiss system to have a double fluorescence system. And maybe the other idea could be to try to observe this conformational change with a molecular point of view. For example, in this paper, with this type of DNA origami, it was possible to observe the kissing with the GTP switch. So maybe if we try to design this type of experiment with our system, it should be possible to observe the conditional kissing interaction with the positive and the negative aptaswitches. Just to finish this talk, I would like to thank my supervisors and also my lab team. And the most important, I would like to thank you for your attention.